Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject Recombinant DNA Technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Dear learners, today's topic is vectors. In this session, you will be learning what is a vector and what are the features of an efficient vector, what are constructed vectors, the types of vectors. Dear learners, let's study about vectors in detail. So, vector is a delivery agent or a cloning vehicle or a carrier which delivers our desired gene from the source to the target organism. You know that you will be isolating the gene from the desired gene from the source, and that gene can't be directly inserted into the target cell. You require the support of a vector, a carrier. So, this particular feature, it is satisfied by the vector. So, it helps in carrying the desired gene from the source to the target organism. In general, this vector is a DNA molecule, and the common vectors which are used in genetic engineering, they are plasmids, bacteriophages, animal virus and plant virus. Coming to the features of an efficient vector, whenever you are talking that this particular thing is a vector, it should be able to satisfy certain features. So, the first one is origin of replication site. It is also called as ORI-OR site or ORI-C site or replicon. So, you can call it by any one of these names. So, this replication site, it contains the genes which help in the independent replication or autonomous replication of the DNA molecule. So, the ORI-OR site should be relaxed in nature so that you will be getting more number of copies of the DNA molecule. So, the replication site or the replicon should be able to replicate independently, or self-autonomous replication is needed. The second thing is small size. The vector should be of small size so that it is very easy to manipulate, because here you are changing the genome of the organism, and also you are inserting the genes in the vector molecule. If it is of small size, then it is easy to manipulate that particular vector. The third point is entry and recovery. So, you will be introducing this vector into the host cell. The host cell, it will be providing the suitable environment for the growth and replication of the vector. So, and once you introduce, and then at the end you will be recovering that one. So, the entry and recovery process, it should be hassle free. So, that thing, it should you should also take care that the particular vector which you are handling, it should be easy to recover from the host cell, and also the entry should also be hassle free. The third thing is polylinker sites or multiple cloning sites, because to insert the DNA, the target DNA or the desired gene into the vector molecule should be first, it should be cut. That is, for example, if it is a plasmid, it is a closed covalent circular molecule, you have to open the circle. So, for that you need a restriction enzyme, digestion is needed, and it should have a recognition site. So, if the vector is having many number of recognition site here and there, it will be difficult, and there is a possibility that the vector DNA will be made into fragments. So, to avoid this one, what you are doing means what you need is this polylinker site or multiple cloning sites. They contain several restriction sites, unique restriction sites which are clustered in one location, so that these particular sites or the regions which contain the restriction sites at one region, they are called as polylinker sites or multiple cloning sites. So these sites, they avoid the breakage of the vector into multiple fragments, just it will be broken at one particular site and you will be getting a linear fragment if it is a plasmid. So restriction enzymes are used to cleave this polylinker site to open the cloning vector without disrupting any other region. So this is also a very important feature. And next, coming to the marker genes. So you are saying that you are inserting the desired gene into the vector. So and then that recombinant vector is introduced into the host cell. So the host cell, if it takes the recombinant vector, it is called as a transformed host cell, and if it is not able and if it is not competent to take the this recombinant vector, those cells are called as this non-transformed cells. So these marker genes, they help in easy detection of the transformed cells. So you know that the transformation frequency is very low and you have to sort or separate the transformed cells from that of the non-transformed cells. Then this marker genes, it helps in the easy detection of this. So we have seen that the vectors are naturally available like this plasmids, then phages, animal virus and plant virus, and these they can be used as vectors, but those vectors, they are not satisfying all the features which we have discussed. That is, some will be having small size and some will be having poor marker genes and some are too large in size to handle. So all the features which we have discussed in the previous slides, they are not satisfied by this natural vectors. So in order to avoid that, this in genetic engineering, the vectors are constructed, or synthetic vectors or constructed vectors are designed. Why? Means the naturally existing vectors do not possess all the efficient features, like if you take PSC 101. So this is the plasmid which is stringent in nature, that is the replicon is it is not this relaxed form. So due to this, multiple copies of the DNA is not produced. And if you take the Col-E1 plasmid, it is also a natural plasmid, but it has poor marker genes. And if you take this RSF2124 plasmid, it is very large size. So our vector should be of small size in order to handle. So these like this, all the features are not exhibited in a single vector molecule. So what you are doing means, so you will be engineering or you will be designing the vector by joining the fragments of DNA taken from different vectors. From each vector, whatever suitable feature is there, that portion of the DNA it is taken, and then all the fragments, they are joined to produce a artificial vector or a constructed vector. So in this manner, if you see this example, this is the constructed plasmid vector. So multiple cloning site, it is taken from one plasmid, then the replicon, it is taken from another vector. The marker genes, it is this particular portion, it is taken from another vector. Like this, different bits of these fragments, they are taken from different vectors, and then they are joined to form a constructed vector. Okay. So depending upon the end product one expects from the vector, these particular vectors, they are classified into majorly into two types. One is the cloning vector and another one is the expression vector. Apart from these two, you will also be studying about shuttle vectors in the coming slides. So coming to the first example, cloning vector. So if our goal is to produce the inserted gene in multiple copies, then you will be selecting a cloning vector. So here the examples are PBR322 and PUC19 vectors. So these cloning vectors are used to multiply the inserted gene in more number of copies. Coming to this PBR 322, this is the that is the polylinker site, which is containing restriction sites for multiple restriction enzyme, and the ampicillin and tetracycline resistance genes, they are the marker genes and origin of replication. In this manner, the cloning vector will be containing all the essential genes, and in this particular polylinker site you will be cutting the DNA and inserting the desired gene, and this gene will be multiplied in many number of copies due to the relaxed replicon. Then coming to the second type, expression vector. So this one, it is used, that is if our goal is to produce a protein in the host, or if you want to express a character in the host, then you will be selecting an expression vector. So in order to express the desired gene, you require regulatory genes like this promoter, operator, then terminator and other genes which are required for the regulation and the expression of this desired gene. So all these genes, they are added in additional to that of the normal genes which you are seeing in the cloning vector. So apart from this selectable marker, then origin of replication, polylinker site, you will be having this regulatory genes in additional in case of the expression vector. Why these regulatory genes are needed means to express the inserted foreign gene, or to produce the character of this gene, you require the this particular regulatory genes. So that is the main purpose of using the expression vector, and the example here is PCMV BLOS. So it is the one which is used to express this particular inserted foreign gene. It is having origin of SV40, origin of replication, then promoter, enhancer to enhance the expression of the gene, inserted gene, then terminator signals to stop the expression, and the common things are the multiple cloning site, then this origin of replication, it will be having it in common with that of the cloning vector. Next, coming to the last type of vectors, shuttle vector. So this shuttle vector, it is used to propagate in two different species. That is, you can propagate this vector both in a prokaryotic cell as well as in a eukaryotic cell. So it will be containing the replicons of both the cells. Suppose let's take that as York vector. So this is the yeast artificial chromosome, it is a shuttle vector. So in order to propagate in bacteria, this one it will be having origin of replication of bacteria and also yeast origin of replication. So when you use this vector in yeast, it also replicates because it is having this yeast origin of replication, and this yeast selectable marker and bacteria selectable marker. Like this, this shuttle vector is having the capacity to replicate or to propagate in two different host cells because they are containing the genes necessary for the propagation in both the cells. And also the particular shuttle vectors, they are used, it can be used both as a cloning vector and also as an expression vector. So if you add the regulatory genes in the shuttle vector, it also starts to express those genes in that host cell, and the prominent vectors which are used are YARCs and BARCs. That is, the BARCs means bacteria artificial chromosome, which generally used for the construction of these genomic libraries, and these vectors are having the capacity to insert large sized DNA molecules. For this reason, they are used in the preparation of these genomic libraries. So in the conclusion, let's recap all the points which we have discussed. One is the vector is the carrier which delivers the desired gene to the target organism, and in a particular organism, the molecule In order to say that it's an efficient vector means it has to follow all the features like a replicon, relaxed replicon, small size, marker genes, polylinker site and regulatory genes in case if it is a expression vector. So, the three types of vectors which we have discussed, the clone desired gene in the multiple copies. For that you will be selecting the cloning vector, and to express the desired gene you will be taking the expression vector, and to satisfy both the features, that is either to clone the gene or to express the desired gene, you will be taking the help of the shuttle vectors, and it is also having the capacity to multiply in two different cell types. Okay. So, with this I conclude the session.
Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.